Hi, welcome to the Field Museum. I'm Greg Mueller. I'm a scientist here at the museum, and I'll be one of your guides on today's Underground Adventure electronic field trip. And I'm joined this morning with Joy Peterson from Best Buy's Children Foundation. Morning, Joy.
Hi.
How are you doing?
Great. Say thank you for inviting me to the foundation's sponsored field trip experience. This is the third in a series of electronic field trips that the Best Buy Children's Foundation has had the honor to sponsor. I'm so excited about today's event. We understand there are more than 800,000 children in the audience this day, and we're going to learn all about soil from across the country. Best Buy got involved because we are excited about cutting-edge technology and how it reinforces education for today's youth. We are involved in educational programs all across the United States where we have over 344 stores. But this is one of the rare opportunities we have to work on a national level. Can you tell me what we're going to do today?
Yes. What we're going to do is we're going to talk about the incredible world of life under the soil. And while we're doing this, we're going to invite people out there in the audience, all you students out there, to call us. There's an 800 number on your screen now, so use that number. You can also email us. And so use either one of these anytime during the program, and we'll get back to your questions. So what we're going to do is talk about the life that exists under soil, how they're interconnected, how we're interconnected as people, that we can't live without these organisms. And to do this, we're going to do two things. We're going to spend some time in our exhibit that's behind us, and then we're also going to go upstairs into our collection area on the third floor and show you some of the real organisms that we've been getting from the Chicago area and from various places around the world. I see you brought your box. Great. So it's full of good Minneapolis soil, correct? Terrific.
Can you tell me what this is all about now? I'm not very smart about plant life. I'll hold this and you explain it.
Okay, well this is a big blue stem, and I'm going to need your help. If you can hold that, we're going to actually keep building this because this is a tall plant. This is the dominant grass in our prairies, and we could keep on going up here. And so this thing is really tall. But what's amazing is we didn't have space for all of this. And if we tip this down a little bit here, Joy, we've got a few of the roots, and you can see the roots down here. But if we had room, we'd have all 8, 10 feet, even more of the roots that go down on the surface. And what we're going to do to get started is we're going to shrink you down using our shrink chamber that's behind here. It's not going to hurt too much. Anyway, we're going to shrink you down and send you down between the roots. And you're going to see all the critters that live down in there among the roots. So if we can just move on, you're going to go behind there. You'll walk through and get shrunk to 100 times smaller than you are right now. Okay, have fun. Good luck. There she goes. She's going to start shrinking. Okay, there she is. She's in the shrink chamber. She's moving her way down. Oh, there she is. She's going to get smaller soon here. Oh, yeah, she's getting smaller. She's getting smaller. And there we go. And when she gets to the other end, there's going to be Petra Seerwald in the exhibit in the microsoil lab that's going to be able to tell us more about that. Petra, are you there?
Yes, I'm here, Greg. Thank you. I'm Petra Seerwald. I'm also a researcher here at the Field Museum. I'm in the Department of Zoology, and my specialty actually are spiders and millipedes. But I also worked a little bit on all the other animal questions when we built this exhibit. But first, sort of the question, why do we go through all this trouble of shrinking down? It really doesn't hurt very much, and most of the time we get the people up to the normal size again. But why do we go through all this trouble? Well, the spaces in the soil are really tiny, and there are thousands and hundreds of thousands of organisms living in these tiny spaces, but because they are so tiny, these spaces, the organisms are also small. So we have been shrinking people down here in this exhibit very successfully 100 times, which also means that our surroundings for us is 100 times larger than life. So we are really incredibly small right now, and everything around us is 100 times bigger than it would be normally. We can imagine that a little bit. If you think of a penny, if you put a penny onto your hand, the penny will about cover sort of the tip of my finger. But if you look at the penny right here behind me, I just hope that thing doesn't fall over because it could easily crush us. So this gives about an idea how small we are. But perhaps we ask Brett, could you come up, please, and measure yourself? Thank you. Go to that, our yardstick. So how tall were you before we shrank you down, Brett?
I was about 4 feet.
About 4 feet. Well, now he is just half an inch. So that is how small he is. Thank you. And all the animals were also shrunk down. And if you look around, some of the animals around here on the wall are still relatively small. Amelia, you knew what were these kind of animals over there on the wall? Those ones?
This is a mite. So is this one.
Yes, right.
And over there is a different kind of a mite.
Okay, yes. Looks very different.
And up there is a springtail.
Right, up there is a springtail. Do we have that on camera? No. Now we have the springtail on camera. An insect without wings, but you can see the 6 legs. Thank you, Emily. Yes, Amelia. We have the, I think this goes to? Yeah, right. So we have the animals here in the exhibit, and these ones are relatively small, although they are already 100 times larger than life. You went through the exhibit before, though. Do you remember other animals that were larger than life? Yes, Mia?
There's the wolf spider.
My favorite.
And I forget the other one's name.
The crawfish.
The crawfish.
Was really big.
And
yes.
They're both really big.
Yeah, they are larger than we are, 100 times as large. What is also larger than life here are these sort of upside down bushes in the soil wall. And Mia, what was that? What was these bushes? Upside down almost, yeah.
These are roots from plants, and they come down into the ground, and they'll from the ground, they'll take up waters and minerals and feed it up to the plants.
And nutrients, right. That's what they get out of the soil. And these are actually the roots of the big blue stem. So you could see this little fuzz that Greg had at the bottom of the blue stem plant before. But now this is, these are the roots here. And you can see the soil is pretty much covered and full of these roots. And it's right, the roots take up nutrients and water for the plant. But what do the roots do for the soil? Do you remember that? I think we talked about that sometime before, what the roots do for the soil. Yes, yes, Mia.
Well, like when the plants get taken away, the roots leave holes for the soil for it can have air.
Okay, and also the roots hold the soil into place. I understand we have an email. Are there different kind of worms? Well, great question. Of course there are different kind of worms. There are thousands of different species of worms, and every worm species is adapted to a specific habitat, eats particular food, and has a different role in the soil. So there are hundreds and hundreds of different species of worms and very different kinds. We have a couple here in the exhibit. If you look here at these thin threads, these are nematode worms, and they live in the soil. There are so many nematode worms in the soil that if all the earth would be gone, and you would look at the earth from outer space, and you would only have the nematodes, you would still see the continents. There are different species of worms in the soil than elsewhere, yes. There's a mind-boggling number of different species of all kinds of these organisms. And there are researchers that are often specialized in study these different organisms. All right. I think what we were also wanted to look into today was how this precious resource that we have here, soil, is made. Because soil doesn't come just from nothing. It actually takes an awful lot of time, and it's a very important resource on our planet. Without the soil, we would have no plants on the land. We wouldn't live here. So We wanted to head over to Maria in the mud room of the exhibit and look there and see what Maria can tell us about how soil is made. Maria, are you there?
Hi, thanks Petra. My name's Maria Fontana. I work here in the museum in the education department. I'm a special projects manager for the Monsanto Environmental Education Initiative. So I work on a lot of environmental projects here. With me, I have Carly and Sajin and Zach, and they're going to be helping me while we work on the soil. Now, one of the things that I'm in charge of is I work with our soil adventure mobiles, and those are trucks that go out to schools, and the presenters work with the students in the schools to talk about the soil right where they are. So usually when we go out to a school, we take a soil sample from the school. But today, as you can see, we're in the mud room of the underground exhibition. This is the place where people can do all sorts of hands-on things, and that's why we'll be doing hands-on things here with soil. The first thing that I'd like to show you is some of the boxes that have been sent to us from around the country. We've got boxes of soil sent from all over. This one, I hope you can tell right away, is from Chicago. And we have another one here from Las Cruces, Mexico. Now, both of these boxes, the story behind them is that the students were able to collect the soil near their school. There were different types of trees or larger plants growing there, but not a lot of smaller plants. We also have soil from New York. Can you help me, Carly? From New York and Fort Wayne, Indiana. These students were able to collect their soil in the yards near their schools. I believe that there was a mulberry tree near where we collected the soil in Fort Wayne, Indiana. Inside each of these boxes is soil collected from near the schools. We also have soil from Texas and from another part of New York. And as you can see, the students have really done a nice job on decorating their boxes. These came through the mail to Ball State University, and actually a couple of them were sent here to the Field Museum. And let me show you just a couple more. This box we have here is decorated actually by the people that are standing here with me at the table. This box is from Burris Elementary School, and the soil was collected from the yard near their school. And this box is from the Field Museum. So we've collected soil from right outside our doors here at the Field Museum. The last boxes that I'll show you are from New York and Connecticut. So as you can see, we really did have people send soil here from all over the country, from the East Coast to the West Coast. Okay, so you guys, let's look at the soils that we have here. We've taken all the soil out of the boxes and we've placed it on all of these plates. What I'd like for you to do for me now is I'd like for you to look at the soil, and I want you to touch it and observe it, maybe listen to it, and tell me some things that you notice about your soils. Sajin, let's start with you. How about? Tell me a little bit about the soil that's in front of you. Where's this soil from?
It's Texas.
Okay, and tell me a little bit about it. What do you notice about it?
It's kind of tan.
And what are
Rough.
the, maybe you can help me with this. What are the things that make up soil?
Sand, silt, and clay.
Sand, silt, and clay. I thought there was one other thing. Is there one other thing, you guys? Carly?
Living organism.
Living organisms like
Yeah.
organic matter, things that die? Okay, so definitely we'd have things that are dead in there because that would make the soil more fertile. So what does yours look a lot like? Sand, silt, or clay?
Sand.
A lot of sand. Can you see the individual particles in it?
Yeah.
Excellent. Zach, what about your soil? Where's your soil from?
Indiana.
Okay, tell me a little bit about your soil.
It's hard. It's stuck together, and
What does that
It's-
tell you? That it's stuck together?
it's clay.
A lot of clay in that soil? And what does that mean about growing things in that soil? Do you think it'd be easy or hard?
Hard.
Okay. Does anybody have an idea why? Looking, you guys, Carly and Sajin, look at this soil. Why would this be hard to grow plants in?
It doesn't have much sand.
Okay, it doesn't have much sand in it. Good. Carly, tell me about your soil really quickly.
Mine is more bigger and crunchier than this sand right here.
Okay, and compared to these, it's a lot darker, isn't it?
Yeah.
Yeah, and it's a lot smoother? Okay, what we're going to do now, could you guys help me out? Could you move these 3 soils down to the end of the table? A lot of times what soil scientists do is they will make a soil shake with the soil that they have. Zach, this is for you. And then Sajin, this is for you. And Carly, this, why don't you come on down here, and this is for you. We've taken 3 different soils. The soil on this end is from New Mexico. Sajin will be working with Fort Wayne, Indiana, and Carly will be working with soil from New York. What we've done is filled our baby food jars with 2/3 full of water, and now what the kids are going to do is fill the rest of it with the soil. And once they've filled it, you guys go ahead and do it. Fill them with soil. And I'm going to help you out just a little bit so we can do it quickly. Once we've filled them with soil, we're going to shake them up really hard so that we don't have any lumps left in the soil. And what scientists will then do is look at how much sand, silt, and clay are in the soil. Okay, once you guys have this done, can we have you shake them up really hard? You need some help there? Okay. How are you doing? Okay. Will you shake this up really hard? And we did this yesterday. We made soil shakes from a lot of the soils yesterday. And so we're going to show you what it looks like when you first start to make the soil shake and then when you're finished with the soil shake. Make sure you have to make sure the lids are on really tight or it's a really messy business. You got that? Okay, now could you guys hold up the soil shakes you just made and the ones that we made yesterday? Can you get it? Oops, sorry. Looking here at Carly's, we can see that the sand, silt, and clay have already separated out on the one that we did the other day, and we've got the organic matter floating around on the top. That's good. That's good. Excellent. Here, go ahead and hold these up. Okay. And that's how you make a soil shake, and that's how you can find out in your own backyard or in your own school how much sand, silt, and clay there is in your soil. And I understand that there's an email. Why is New Mexico soil so sandy? New Mexico soil is so sandy because New Mexico is situated in one of the deserts that we have in the United States. We have another email. How long does it take to make an inch of soil? Generally, it can take anywhere from 100 to 1,000 years to make 1 inch of soil that actually is a usable, good soil. Okay, we're going to go off to Pat and Greg now.
Hi, it's Greg Mueller again. We're up here now in the collection area of the museum, and I'm joined by my colleague, Pat Leacock. How you doing, Pat?
Good. How are you doing, Greg?
Good.
We're up here in one of our research collections areas. If you can get to see down there, we've got many cabinets of specimens, and we've got a few examples here from both the zoology department, such as the several million insect collections they have, and the botany department, which has 2.5 million plant specimens. And we also have a collection of fungi, my favorite organism.
Mine too. What do we got in there?
And you can see we house these a little bit differently in the boxes here.
This one for you. So what do we do with all these things?
Yeah, you might wonder why we have all these specimens. Well, one reason is for education purposes, so people can visit the museum, learn what these different organisms look like and how they live and where they grow. Another reason is for loans. We borrow material from around the world and also loan out our material to different institutions around the world.
So it's like a library.
Yeah, it's a library. It's a specimen library.
Specimen library, and so everybody can do research and study these things. That's great. We've got projects. I know I work in Costa Rica and I work in China, but we also work in Chicago.
Right,
That's
we have
where
several
you
sites
really
in
work,
Chicago.
right?
One is the Swallow Cliff Project. And we'll go down here and see what's going on with that.
Okay.
And meet some of the students from Burris Elementary and Harold Washington.
Oh, great.
Hi, Jim. How's it going?
Hi, Pat. Hi, Greg.
Hey, Jim.
What's going on down here?
We're working down here with some of the insects that are found out in the Swallow Cliff Forest Preserve District on the suburbs of Chicago. I'm Jim Loudren. I'm work in the insect department here at the Field Museum, and I'm a collections assistant. About 2 weeks ago, Pat and some of the students from Harold Washington School went out to Swallow Cliff and did some collecting.
With you too.
And I was also there. And we have some video of what we collected. Here are the kids, they're sweep netting. They're collecting the insects that are in the grasses and plants that are growing at Swallow Cliff. These are cerambycid or longhorn beetle larvae. Now we're collecting litter samples. And what we do is we collect leaves and rotting logs, and we throw them in the sifting bag. And we just collect a whole bunch of material off the ground. We throw it in. Once the top part of the bag is full, we shake the bag, and this takes the small parts of organic matter and the insects and drops them down through a screen down into the bottom of the bag. We then take the sample, put it in a collection bag so we can bring it back to the museum. Here we're taking a live insect out of a collecting jar and putting it in a specimen jar in order to bring it back to Harold Washington School to look at and identify. Here we're taking the sweep net sample and putting it in a jar, and we're pouring alcohol in. And the alcohol preserves the sample so that we can bring it back to the museum again and study it and see what's in here. At this point, some of the students are looking into the jar of matter and seeing what kind of insects are in there. And we know there's some grasshoppers and some leafhoppers and things that we can see in there. There's also some plant hoppers and some caterpillars, some spiders, and a whole bunch of different insects are in there. Okay. Oh, here we are back up in the museum collection area.
Hey, Jim, I just heard we got a couple of email questions. One of them is, how many types of insects are there?
Oh, there are a couple of million different types of insects, different species. Only a small percentage have actually been identified. Most of them are found in the rainforest, and they've never even been seen or found at this time or identified.
And of these couple of million, the other email question is, what's the most dangerous?
The most dangerous would probably be the scorpions and millipedes, because they do have poison and they can bite.
Okay, well, Jim, why don't you show us what you got?
Okay.
And I'll just stand and watch.
Thank you, Greg.
Sure.
Okay, here we have a Berlese funnel, and when we come back to the museum, we put the sample in the Berlese funnel. Here you go, Charisse. We'll let you pour some of the leaf litter into the funnel. I'll give you a hand here. Just go ahead and pour. Go ahead and let it go. There you go. Okay, that's good. Okay. Okay, Mika, now you'll spread it around. Okay, that's good. Now what we'll do is we'll take and put a jar of alcohol down at the bottom of the funnel, and the insects will fall into the alcohol. Okay, then we'll take the cover, and the cover has a 100-watt light bulb, and the 100-watt light bulb creates heat. Why do we create the heat in the funnel?
You create the heat in the funnel so that it could irritate the bug skin so that they could fall down, go down to the bottom and fall into the jar of alcohol.
Right. When the insects get hot, through evaporation, they lose the water in their bodies. And of course, insects are mostly water, just like humans are. So they're always trying to crawl away from the heat, and they crawl down through the litter, down through the mesh, and fall down the funnel into the alcohol. Then we take and we turn the light on. That creates the heat. And in a very short while, the insects will start crawling down, and they will fall into the alcohol. And in the alcohol, they will drown, so we can take the specimens, put them under a microscope and study them, find out what kind of insects there are, what kind of other invertebrates there are, and just do a lot of different types of research on them. Now we're going to go over to the microscope and look at some of the different invertebrates that we collected out at Swallow Cliff. Okay. Here we have our first sample under the microscope, and you can see that there's a penny under here to show you how small some of these insects really are. Who knows what kind of insects are in here now?
Pseudoscorpions.
Right, these are pseudoscorpions. They're really, really small. You can see that the biggest one is only about the size of one letter on the word one on a penny. These insects crawl around and hitch rides on other bigger insects to move from place to place, and they eat decaying material. Okay, now we'll put another sample on. Okay, does anybody know what these guys are?
Staphylococcus beetles.
Right, these are staphylinid beetles or rove beetles. These beetles also are decomposers. They eat, some of them eat plant material, some of them eat dead animal material, and again, they turn this material into very fertile soil. Okay, next sample. Let's see what we've got in here. Okay.
Spiders.
Right, these are spiders. Spiders, of course, these spiders are hunters. They crawl around and hunt for their prey. A lot of spiders spin webs, and they spin the webs to catch their prey. Okay, next. Let's see what else we've got in here.
Those are centipedes.
Right, these are centipedes. These guys crawl around, and these are hunters. Centipedes actually do have venom to kill other small insects. And they're hunters, and they kill the insects and eat the insects. Okay, we've got one more. Okay, Penny's moving out of frame. There we go. Okay. Anybody know what these guys are?
Mites.
Right, these are mites. These are very, very small critters, and they live on different animals. They ride around on animals, and they eat dander and things like that from the animals. Even humans have mites on them. They have, in your eyebrows you have mites, and they eat the dead skin. They keep the dead skin off of you so that it helps you stay clean. Okay, when we went out to Swallow Cliff, this is the sample that we collected out at Swallow Cliff. And all that litter that we collected, when it went through the funnel, came down to this. Anybody have any idea how many insects might be in here? Just a guess, a wild guess.
600.
That's pretty close. There's probably about 500 or 600 different invertebrates in here. All the different ones we looked at, the Pseudoscorpions, the Millipedes, the Centipedes, all those different insects are in here. Okay, now we've got one more thing that's kind of interesting to look at here, if we can find one of them. Oh, here he is. Okay, here you go. Everybody knows what that is, right? That's a Cerambycid larva, a longhorn beetle. That's the larva. Just go ahead and pick them up. Go ahead and hold it up to the camera there. And this is the actual adult Cerambycid or longhorn beetle. Most longhorn beetles are wood eaters. They can become pests, but most Cerambycids or longhorn beetles do have lots of predators that keep their numbers down, so they really, really don't become a big problem. Oh, and I notice here on these pieces of wood, there's some really cool fungus growing here. Let's give this to Pat and see if he can tell us a little bit more about it.
Well, that's great,
There you
Jim.
go, Pat.
This
Thank
is
you.
this is the kind of stuff I study, fungi. They're sort of weird critters in between animals and plants. They're not quite either type. And fungi have a lot of different roles in the soil and wood. And this is actually the main part of the fungus. This is the growing part. This is where it's decomposing the wood, getting food for itself. It can't make its own food, so it's breaking down the cellulose and other wood components. So it's sort of analogous to the leaves on an oak tree, because the leaves are providing the food for the oak tree, whereas the vegetative mycelium is providing the food for the fungus. Michael, why don't you go ahead and open that box, see what we got out at the Swallow Cliff a couple weeks ago. The stuff we got out on our field trip, it was mostly wood rot fungi. That's what we found late part of October. And these are all different kinds of fungi that are breaking the wood down and recycling the wood particles into the soil. And wood is one of the important components of forest soil. It helps retain moisture in the soil. And these fungi that we see here are actually just the reproductive part of the fungus. So the mycelium was the growing part. This is the part that's producing spores. And here we've got, what are these guys?
Acorns.
These are acorns from the oak tree. This is the reproductive, or the seed, from the tree. So it's analogous to the fruit body or the mushroom of the fungus. And fungi produce spores, which are microscopic little particles that serve like the seeds. And they produce those on different kinds of surface areas. And we've got a couple examples here. And we've got close-ups here, I think, of these. This shows a polypore. It's many different pores or chambers underneath where the spores are produced. Next one shows this yellow powdery one, or that's a gray stearium, one that the students identified. And that's got a gray smooth surface for producing spores. And the next one, okay, that was it. So that's different ways to produce spores for fungi that are growing on wood. And these These are wind dispersed, so the wind carries the spores away to new locations. And we have an email question. Do plants feed on fungi? That's a good question. Most people know that fungi feed on plants. There's a lot of parasitic fungi. But there are a few plants that utilize fungi for food, and one of those is the Indian pipe. It's a little white, colorless plant, and it's actually getting its food from a fungus that is growing with a tree, and Greg's going to mention Mycorrhizae later. So the plant is getting its food through the fungus from the tree, so it's a three-way partnership there. And we've got a couple things here that are, instead of wood decomposers, they're decomposing stuff in the soil. And you guys remember these from out at Swallow Cliff, right?
Yeah.
What are these?
Giant puffballs.
They're puffballs. And these are dispersed when raindrops or things hit the spore chamber, and you get a little cloud of spores that get puffed and carried off in the wind. Yeah, Michael.
Can these grow in the trees in your backyard?
Yep. If you've got dead wood around in your yard, you'll find quite a variety of these. These are fairly common wherever you find wood in the Chicago area. We've got a giant puffball here, and we had a shoebox from New Mexico where it's real sandy. So we've got this fellow from the desert. It's a desert stalk puffball, and you had an idea, Darlisa, why this might have such a long stem.
I think it has such a long stem. Because it had nice roots that were really rooted into the ground.
Right. So this fungus is down deep in the soil because there's more water and nutrients farther down in the sandy soil, and this digests the dead plant roots. So the stem has to come way up to the surface where the fruiting body is made. One cautionary note is if you're out enjoying fungi, don't eat them. And you guys remember this one?
Jack-o'-lantern.
That's a jack-o'-lantern mushroom, and it's one of the common poisonous mushrooms we have. So you basically don't want to eat any fungi you found out in the woods. Stick to the ones you find in the store because they're the tried and true ones. We have an email question. How do you know a mushroom's edible? That's a good question we get a lot. And actually, there's no way to tell. It's only through recorded history. People eat it and write down whether they got sick or not. And so it's only through centuries and centuries of people utilizing fungi as food that we know which are safe edibles and which are not so good. This is an example of a deadly mushroom, one of the 6 or so deadly mushrooms we have here. This is an amanita. And one characteristic of an amanita is this swollen base at the bottom. We have a close-up of that. There's the, shows you 2 fruit bodies, 2 mushrooms. It's an all-white mushroom, and at the very base, there's a swollen, enlarged area that's a vulva or a cup at the base, and that's one way to identify an amanita. But the trick is that can often be underground, so you want to be sure and dig that up when you collect a mushroom. Okay, we're going to go down to Francie, back down an underground adventure, to have her show us how she incorporated things like the mycelium and the fungi and the insects into the exhibit. Francie?
Thanks, Pat. Hi, I'm Francie Moraski-Stotz, and I'm in the exhibits department here at the museum, and I was the leader of the team that created Underground Adventure. When we create an exhibit like this, we work very closely with scientists like Pat and Greg and Petra to make sure that everything we're doing is accurate, and that's particularly important when we have an environment like this that we've had to create. This particular thing I'm standing in front of we call the Root Rot Hotspot, and it has that fungi, et cetera, that Pat was just talking about. What you're looking at is a dead root, and all the white things growing on it are fungi, and that fungi is rotting the root. Now, that's not where the story ends, because there's little things like these mites down here that are eating the fungi. So they're grazing on the fungi while the fungus is rotting the root. And then bigger things like the beetle down low or the pseudoscorpion up high are predators, and they're hunting the small things that are grazing on the fungus. So we have a whole food chain modeled here to help people see how important decomposition is underground. Now, if you remember what Jim showed you with the pseudoscorpion on the penny, next to the penny, you realize we have to model some very, very small things here. And to do that, we took photographs of the specimens using an electron microscope. And that's what lets us get some of the fine detail. And we have a good example of that over here in the ants. You can see this is an electron microscope photograph of one of our ants. And it lets you see all the fine detail, how the antennae connect, the little bristles on the face, the eyes, the bumps. And with that, our model makers can make our models very, very accurate. So now let's look at our real ants. Why do you think we included ants in here? Well, we're all familiar with anthills in the soil, but you may not really realize what's happening in those anthills. If you look at one ant in particular in our ant colony, it's got a chunk of soil in its mouth. And what it's doing is, as it's digging, it's bringing up particles from underground, from deep, and other ants are bringing plant material from down above, and so they bring organic material down, minerals up, and that actually helps make the soil richer, and it's mixing the soil and actually helps speed new soil formation. So that's really why anthills and ants are so important. Now, another organism for which we use electron microscope photos to model is the wolf spider. And you can see a photo of the wolf spider on your screen. You can see it lets us look at all the fine hairs or setae on the face, and all the round things are eyes. And the eyes on this wolf spider, there's 8 of them. So we used this photograph to make sure the wolf spider was very accurate. Now, of course, those electron microscope photos don't show color. So to actually get the color of this, we had to use a real specimen. And the color that we duplicated was using a little bit of brown hair, a little bit of gray hair, and actually a little bit of purple. Now, it wasn't just the look of the spider that we had to get accurate. With these animatronics, the ones that are moving, we had to get the movements accurate as well. And so if you take a look at how this spider is moving, it's not just moving its leg, it's moving its jaws in and out. And that helps us really see how this spider feeds. Now, it's feeding on a beetle grub. And as it feeds on this beetle grub, it's a hunter, and so it hunts things underground like the beetle grub, but it also hunts many things above ground. So showing this movement helps us emphasize what the spider is doing underground. Okay, now I'm standing next to some clover roots. And these clover roots form a very interesting association. The lumps you see on these are nodules, and they're filled with a bacteria called rhizobia. And these bacteria are too small to even see at this enlarged scale. And this is a very interesting partnership among a plant and a bacteria. And to talk a little more about that, we're going to go back upstairs to Greg in the research labs. Hi, Greg.
Hey, Francie, how you doing?
Fine, how are you?
Doing well. Yeah, you're right. Those bacteria that's in the rhizobia nodules there are so small, I don't even have any specimens of those. But, you know, this is a type of association between a plant and another organism. And I'm going to ask if anybody knows what that happens when you got 2 organisms living together, benefiting each other. What's that called?
That's called symbiosis.
A symbiosis, you got it. Is that the only symbiosis we got down there? We'll have to come back to that. Just here we got some email. Could I have my email? No email. Okay, then we're going to talk about another symbiosis we have that Francie was showing, and that was on this oak log. And we've got mushrooms here. This is a fungus that's growing above ground, but if we went below ground, we'd have mycelium and then this stuff. Do you know what this is here under this microscope?
Mycorrhizae.
Mycorrhizae. Do you know what's a mycorrhizae then, if I just didn't help you with that one, huh? Oh, haven't seen the oak root. Okay, so, yeah.
Mycorrhizae is something that grows along with the root, because if mycorrhizae wasn't along with the root, none of them could survive without each other.
Excellent, excellent. Hey, can we go back to Francie and see that oak mycorrhizae that's down there in the exhibit? Francie, you still there? Oh, there we go.
Yes, I'm here.
Okay, you see that big root? That's a combination of the tree root and the fungus. Remember, this mycelium grows out away from the tree, and so it brings in water and nutrients to the tree. And what does a tree do? What do plants do? Anybody of you guys know that?
Photosynthesis.
Photosynthesis. What's photosynthesis?
That's how they create their food.
They make food. Okay, Francie, so this is going to be an idea again of above and below ground, right?
That's right. this whole area shows not just relationships between organisms, but between the below ground world and the above ground world, because you can't separate them. Without what's happening below ground, what's happening above ground would never exist.
That's great. So yeah, we got this combination above and below. Thanks, Francie. Let's see, what else do we got up here? So we've been talking about symbiosis. We're talking about relationships between bacteria in plants and fungi in plants. But not all fungi are beneficial for plants. Some are actually going to kill them. And one of them that we have here is this guy. This is the honey mushroom. And the honey mushroom is actually one of the largest organisms on Earth. It's called the humongous fungus, is one of its common names, because they found an individual that was 37 acres in diameter and was over 1,500 years old. So it was like one of the oldest and biggest things. But what do you think if we went up there? This was up in Minnesota, Michigan. If we went up to the Upper Peninsula of Michigan and was looking for this, do you know what we'd find? Anybody want to make a guess what we'd find?
A massive number of them?
Yeah, a massive number of these guys, but most of it would be underground. So what we'd have is a couple of clumps of mushrooms over here, a couple clumps of mushrooms over there, because it's that mycelium, remember, that's actually growing underground. And so this is the part that extends and lives for the 1,500 years. This just comes up here and there. But this isn't the only big mushroom. This is the largest individual we know, but I've seen a bigger mushroom once. Found this thing in Costa Rica. And I think I was talking about this. You remember how big this thing was?
It was 3 feet in diameter and weighed 18 pounds.
3 feet in diameter. I know kids smaller than that. I mean, that's humongous. That's really big. And I think we have a picture of it when it was collected in the field. One of my assistants down in Costa Rica, there she is. And you can see how big this is.
Excuse me, Greg.
Yeah.
I want us to know if the type of mushrooms like that, are they poisonous?
Well, that's a good question. No one has eaten this one, so we don't know for sure. But when I collected it, it had a really strong odor, and it didn't smell good, so I don't think it would be good. One of the things we're doing in Costa Rica is we're looking for new medicines, and for anti-cancer, anti-AIDS, and so we're screening this now to see if it has any new compounds that might be useful for helping people. And that's one of the things about fungi, is that a lot of these make new compounds and can be used in medicines. The other thing that's interesting about this mushroom is it was growing out of an ant nest, a leafcutter ant nest. And to talk more about ants, I'm going to throw it over to Petra and hear her talk about spiders and ants and whatever other good crawly critter things that she wants to talk about. Okay, Petra.
Yes, hi, this is Petra again. We are up in the collection area. Thank you, Greg. And I was working on all of the animals in the exhibit, or at least kind of researched the content and worked on that. One of the groups that Greg was talking about were the ants, and we do have in our collections in the museum, of course, also ants in the collection. And Greg mentioned the leafcutter ants. Leafcutter ants are incredibly interesting ants, and they are great fungus farmers. All ants live in colonies, and I think you have seen perhaps some of the recent sort of bug and ant movies and know about that. These leafcutter ants cut the leaves from the tree. Workers in the nest, in underground nests, chew up the leaf and then the leaves, and then plant fungus on these on the vegetable paste in a way, and harvest these fungi for food. I hear there's an email. Can you give me that email right now? Oh, that is a question. Is it safe to eat some insects? Yes, I think it is safe to eat some insects. Actually, I had some. I had a barbecued cricket on a celery stick with peanut butter. It basically tasted like, well, crunchy peanut butter. There are insects that can be eaten. That's true. There are even some people who eat spiders, but that is a relatively rare occasion. We do eat another kind of arthropod, though. We all eat a lot of shrimps and crabs. So that are about the only arthropods we really eat on a regular basis. But we do eat shrimps, yeah, but not insects or spiders. That is, although you can eat some, yes, can happen. Okay, there's not just, we were talking about ants before, and there's not just one kind of ant on this planet. We talked earlier about ants. How many species of ants were there on this planet? Do you remember that?
There are over 9,000.
There are over 9,000 different species of ants. They all live on land. Most of them have underground nests, and so they are very important for the soil by bringing soil particles up to the surface, creating nests, but also bringing organic material down into the soil. My favorite animals, and the animals I know most about, and sort of I like to talk about them best of all, of course, are spiders and millipedes. And we do have, for example, in the Swallow Cliff project, where we looked into the fungi, we also find a lot of millipedes. And I would like to walk over there and perhaps get one of the millipedes out of its container, and we find it. Who wants to hold the millipede? You want to hold the millipede? Okay. So these millipedes are also arthropods, like shrimps and insects and spiders. This one is alive, although it hasn't actually, I guess it didn't sleep well enough last night. They are more active actually at night, and it may be sleepy now today during the day. They are very important in the soil because they are decomposers. They work hand in hand with Greg's and Pat's organisms, the fungi. They help to break down leaves and wood logs, so that the nutrients can be recycled back into the soil, and the soil sort of holds then the nutrients like it's sort of the world's fridge in a way. It holds these nutrients for the other plants to grow and use the nutrients again. This one is really sleepy today. Okay, we have an image of a millipede right now, I think, on the still. This one is sleepy. Let's look at some of the millipedes, how large they can get, and also animals that they are most often confused with. But of course, my experts here don't confuse them, right? Which ones here are the millipedes, and which ones are the, I think, perhaps let Mika say that. Which one? Okay.
These are the centipedes, and the rest are millipedes.
Right, and the big difference between centipede and millipedes are that the centipedes are hunters. They are hunters like my spiders are. But the millipedes, and you can see how big some of these can get. This one, for example, is from southern Illinois. So that's not that far away from Chicago. So this is a pretty good sized millipede. So if you encounter a centipede, don't get involved in it. They bite, whereas millipedes often are, especially the millipedes in the area around here, are really harmless animals that help decompose the soil. The T-Rex of underground, that's of course I think the last group I would like to talk about, and that are spiders. I have to talk about spiders. If you think that the spider down there in the underground exhibit is big, yeah, but it's enlarged. If you compare it to the penny, it's still smaller than the penny. Here we have one of our local sort of homey wolf spiders, and I put a penny right next to it, and you can see that this wolf spider is bigger, much bigger than this penny. All spiders are predators, so they are the supreme hunters down there in the underground, and hunt for all kinds of small insects for food. I think we have. We have an email. Okay, thank you. Can you give me the email, please? Well, that's a very good question. The question was, are there more poisonous spider species than insect species? Yes, I would think in a sense, because all spiders, except for one small family, has poison. That's how they catch their prey. That's how they make a living. They catch their prey by paralyzing it, by injecting poison, and but for most of the spiders, the poison is not active on us at all. So And here we have another email. What's the largest spider in the world? Okay, the largest spider in the world is a tarantula, a South American tarantula, often called the bird-eating spider or Goliath spider. It is about, well, how big it is. It weighs about as much as one stick of margarine, if you can imagine that. That's not terribly heavy. But if with the legs stretched out, it fits nicely on a huge dinner plate. And relatives of these are actually eaten by South American native Indians, especially in the northern region. So these are quite big spiders. Sometimes you may be able to see some of these big spiders in zoos or also in museums. And I hear we have one more email. Yes? That is a good question, and it's very hard to tell. The question was, how many spiders live underground? If we consider the number of species of spiders that live underground, the answer is we really don't know. Because whenever we dig up a soil sample and sort the spiders and identify them, about 40%, that's a little bit less than half of the spiders, are actually new species. Species we had never heard about, we have never read about, that nobody has ever seen and written about. So we do have this enormous number of new species of spiders. One of the stills that you can right now see on your monitors is sort of the business end of the spider. That's where the fangs are. And that's what the spider uses to inject the poison into the victim. Well, they usually just paralyze their victims. And that's how they catch their prey. I think we are ready to go down to Jim, looking at the soil samples and of the insects that you found in the boxes that you send in. Jim, are you there?
Yeah, I'm down here.
Okay.
Hi, this is Jim. We're back down in the exhibit with the students from Burris High School, or Burris Grade School in Muncie, Indiana. We're going to talk about the interaction of soil with humans and what happens with the soil. As you remember, we had the shoe boxes of soil and we ran them through the Berlese funnel like we did the Swallow Cliff samples. But there really wasn't very much in those. Can anybody tell me why there wasn't much in those samples? Go ahead.
Because when they build buildings, they take the soil that has been there away and they build the buildings, and then afterwards they put in different kinds of soil that doesn't have as much as living things in them.
Right, right. I understand we have an email. Someone would like to know if worms soften soil. Yeah, when worms crawl through the soil, they do break it apart and leave spaces in the soil, which does make the soil not really softer, but fluffier, so that there's space in the soil for air and water to accumulate. Okay. Yeah, and like you were saying, when they do build new buildings, they do scrape away the soil. They bring new soil, or not new soil, but established soil in and put it where the old soil was after they put the building up and poured the concrete and done all that kind of things. And this unestablished soil really doesn't have much in the way of organic matter or living critters in it. Very few invertebrates or anything like that. And then when they plant things on this soil, what they plant really is what? Grass, just grass. Well, that's only one species of plant. And there aren't very many things that eat plants, that species of plant, the grass. But then if you go out to Swallow Cliff, you find all kinds of different plants. Can anybody have any idea how many there might be out there? Go ahead.
Maybe 100 of them? Maybe 100 of them?
Oh, at least 100, maybe more.
Say 200?
Yeah, probably 200. So with that, all those different types of plants out there, there's lots of things for the invertebrates to eat. Okay, who has their hand up? Okay, go ahead.
I think about 3,500.
There could very easily be 350 different species of plants out at Swallow Cliff. And the more plants there are, the more different types of insects and invertebrates there are. I guess that's about all we have. That's about all we have down here. So I guess we're going to go back up to Greg now, up in the collections area.
We're back up here upstairs, and we're just going to wrap things up. You got any other questions, guys?
Well, you and Petra were two of the major inputs for the creation of the exhibit. What were the major themes?
I don't know.
Well, we wanted to show how important soil is, and we wanted to show different kinds of organisms that people are less familiar with, and how important they are to keep the soil alive, and how much we depend on the soil. And we featured your specimens, your critters prominently, and I think they truly deserve it. Fungi is a group of organisms hardly anybody knows much about it, and I have two specialists right here next to me.
Right. Okay, well, we're going to wrap things up here, and we want to just say thanks to our wonderful students from Burris and from Harold Washington, and thanks to Best Buy Children Foundation, to Ball State for their wonderful technical help, and from Field Museum. And from now, we're just going to say goodbye and hope you all had a good time. You know you're going to look at soil differently the next time you're outside.
I hope so.
Okay, bye now, everybody.
Bye-bye.